slide, and then I think I will ask different questions. On slide, so just have a look on the first one, because I don't remember exactly what I wanted to.
The result?
Okay, no, just the first one first. Yes, you mentioned here that if the rev are not inserted into the nucleus, the mRNA, and you show this one. This one is the fully spliced, the magenta. One? Yeah, the magenta is the fully spliced. This one is, as in the cell, exported to the cytoplasm, and from this fully spliced, we have the translation of REV. The mRNA that are not exported if REV cannot be imported into the nucleus is partially spliced or unspliced mRNA. And do you know why?
Why what?
Why this
is containing the intron?
And only intron? Why REV binds?
It's by with the CR1 on the.
So first REV recognize RRE sequence. So REV recognize a specific sequence which is located into an intronic part. Okay, and it's not the fact that it contains intron. The fact that it contains intron will usually block the export, and this kind of mRNA are destroyed in the nucleus of the cell. But the fact that REV recognize this small sequence RRE located in the intronic sequence, then it binds to it in multimer, and then recruit the CREM1 and RAN-GTP protein to export them. So when you have to show it's this one, to export them, and from this kind of mRNA, this allow the synthesis of new viral particle. So it just change and be consistent with what you say. mRNA exported are this one in purple, and fully spliced mRNA is in magenta, and okay. So number okay. So before questions, I think slide 10. I think 1D is missing, closed. Open and closed. So is it already in, can you correct it directly?
Oops.
Why did you remove the confidence?
Because I don't have enough time to.
You don't have to speak, but just have a look. Because you have to take care about the fact that you can just mention, even if, so you mentioned at one point that the two configuration have a high confidence. So for that, it can be good to have the PAE plot to see it, and then we have to understand that the position of the N-terminal tail, the fact that is unfolded, cannot be really precise. Okay, so it's probably moving, but you can just say, even if the IDR part has not a high confidence, we wanted to test the impact of. Okay, but I think it can be good to add it again in close. You don't. It should not be very big, but just put it like this. You understand that it has been done. 13. I think there is the legend of the Dmax. You have to put dash line for OD delta 14 plus and minus rev. So the graph is okay, but the legend is not okay.
Yeah.
So 16, I think, or 15. Either, yeah, from the beginning, this part. I think it's a bit complicated to understand. I think you have to take care about the fact that you didn't. So so the blue. So the first one is in red, glue. So why this one you don't have a cross here?
I forget.
Yeah.
Yeah, I forget it's
this.
Yeah.
Okay, so you have to combine them, and again here you have one arginine that is not. So yeah, you have to. Yeah. Otherwise, it's a bit complicated to understand. And I think you have to simplify a bit the message. First, we have a map with the interaction. We have the interaction map. We observe that some residue of each monomer are involved. 3 of them involved in salt bridge, the other in close contact. So we first decide to mutate it, the first set of residue in red, that's destroying the salt bridge, but also some of the close contact. And then, as it was, I didn't show the result, but as it was not sufficient to break the interaction, we add a second set of experiment involving the Q15 and the glutamate 36, and we see that these interactions are at the 2 opposite sides of rev, and I will show you just the combination of all, as we didn't see, and so you in that case you can conclude, because I think it's a bit it's
hard to see.
It's not hard to see, but it's the information that you give are not so easy to follow you. So you have really to again say we tested the interaction by native gel and by size exclusion chromatography. So I think it should be mentioned sec here and not fraction. Here you can put the fraction here. And there is something weird because here you have the maximum of the peak is 4, so it seems to be a little bit shifted. And here for the we expect to have something on fraction 8, 9, 10, and you have something here 6, 7, 8. So I think it's not so easy. So I don't know what we have to do. Because
I will fix a bit too.
I think if you remove, so you can let the fractions like this and do it. I don't know, and remove the number perhaps. And if there is any question, you say that's true that the fraction, we are really a tiny colon and the fraction size are quite low, so sometimes it shifts. It's for that it's really important to have a look on the gel. So yeah, just mention because here you mentioned the technique and not for this one, so I think it's important. And then slide 20, I think again 120, the last one
20.
20. One S is missing. Rev binds. And perhaps I don't know if you can, because you switch from OD, HBD, OD, HBD, and I think for the
compare it?
No, it's to just put on one part of, no, not rev,
sorry.
Binds, yeah. It's perhaps you can have a little draw, like has you the one that you present, OD, HBD, and RBD, and when you put it on each slide, and put something in gray here. So you are and you just let it like this, just for the people. When you speak about OD, construction and all experiment performed with OD, you can have this one. When you perform experiment with HBD, you can have the same picture, but you put a rectangle in gray with perhaps, I don't know, 50% of transparency.
Yeah.
And like this, you remove like that, and when you present ODHBD, you will have something like this, and without adding any information, oral information, it's just a visualization of the domain that you are working with, and I think, yeah, it can help in the understanding, because it's a lot of different domains, and you switch from the one to another one, and I think the people that didn't read the report can be a bit lost. First 13 residues stabilize without S. Compactness, number 3. Not sure that.
So what I have to say?
No, it just, I think you add an S first. Not sure that the first 13 residues stabilize without S at the end. Where are you? Here. And so about the question, you mentioned on slide 4, so before in the introduction you mentioned that Rev is a 13 kilodalton residue protein, and on slide 4 on your gel it seems to be much higher.
It's about because it's have a disorder path, so it's perhaps
yeah, you can mention that, yes, is it known, so it's 13 kilodalton in size, that's and the apparent molecular weight is more close to 6 to 18 due to the fact that it's highly basic protein, so a lot of positively charged residue, and perhaps intrinsically disorder part, but usually in SDS page analysis, you should have something that is unfolded. So the fact, I think the composition of residue can, and you can say yes, that's true. That is a 13 kilodalton residue with an apparent molecular weight of 18. On slide 5, can you explain why you have 2 peaks in SDS in SEC, whereas you have a reverse NENTA step?
This because of the tab accessibility, because the ODs form the pentamers, so it's a bit hard for tab to access. Is the localization, its location to cut. So we also, so that is why, and
yeah,
and also because the column is saturation, and the where is it?
So you can, when you answer, you can say with the hypothesis that we draw, because it's not you cannot be assertive. That's not the hypothesis. As we have this reverse NENTA step, and we don't, and we still have his OD form, we suspect. So it can be from the fact that we saturate the colon, but even by increasing the colon like this, you can go further. We still have this kind of results. So we suspect that the accessibility of the TEF sequence is not really good with this construction. Okay. Because we don't have the problem with the GST tag. So perhaps the his tag do funny things. Okay. 5 and 6. Yeah. So here, is it? It's the N15 label protein.
Yeah.
So you have to add it because purify NPM1 ODHBD. So it should be N15 NPM1 ODHBD, because and the list of the protein you didn't present the ODHBD alone, so you have only the N50. So should be mentioned protein purification N15. It's the label form of the fusion protein. Oh, if you mention N15, I think, and usually it's 15N, usually. Yes, and can you, yeah, exactly. Okay. And you have really 3 peaks like this?
Yeah. More, actually 4, but I just only run the gels with 3 peaks, so I only show 3.
So the second one is GST, the first one is ODHBD monomer. And in peak 1, it's weird because
maybe it's the uncleaved protein, because we have a band around 100, so
I don't. Yeah, because the if you look the intensity of the peak 2 and peak 1, and the amount of protein that you have on gel, you cannot really understand why you have so huge, that the intensity of the peak is so high, whereas you don't see any protein, not a lot of protein. Do you agree or not?
yeah.
So and again, why? How can you explain that the intensity of the peak 3 is much higher than the peak 2? Whereas, how can you explain that the intensity on sec of the peak 3 is much higher than the peak 2? And on gel, you see the opposite. The amount of protein on peak 2 seems to be higher than on peak 3. How can you explain that?
Because you mean that the peak 3 is much, the intensity much stronger in the gels.
So here, the intensity on gel, the UV peak is much higher than the peak 2. And on gel, you see that the amount of protein in peak 2 looks much higher than on peak 3.
Is there, because when it is composite blue, so it's also depend on the amino acids of the protein. So, and this ODH3D is the contain the H3D, so it's highly H3D is the basic protein, the domain. So the composite blues left these basics, so it's the intensity is much higher than on the gels, but it's not show that.
When you have HBD alone, do you see the intensity of the band of HBD quite high?
With the native,
with HBD alone?
No. Yeah, right.
Okay.
It's
But I think you can mention on more to explain the difference of intensity on UV peak. Is what
the tryptophan,
the aromatic amino acid that we have only a few, no tryptophan and only tyrosine in the sequence of ODHBD, whereas with the GST contains a lot of aromatic residue. Just, I think you have more to focus on that and they say, okay, the peak 2 is not so high due to the amino acid composition of ODHBD, which doesn't contain a lot of aromatic residue. Okay. Okay, so number 12, I think. And it's very good, this one. Yeah, I like it.
12. Here.
Here. So I think you probably have the answer now because we speak a lot of time about that. How can you explain that by mass photometry you have two peaks? And you mentioned that you have a partial dissociation of the complex, whereas on a native gel you see a clear shift of what is your hypothesis about that?
Because the two experiments start at the different concentration ranks. With the native gel, it has a stacking effect, and with the highest local the concentration, it forces the complex to form. And with the mass photometry, we work with the extremely dilution, which means the concentration is lower, so the mutation lowers the affinity. So we can the dilution cause the weaker the complex dissociate.
So why did you didn't use mass photometry to analyze your OD and Rev mutants?
I will. I also
it's
did.
it's planned, but the accessibility of the machine is not always easy. The machine is book a lot, and this kind of experiment, sex and native, we can do it on a, but that's what we would like to do. Okay. On 14. So just before the SEC, you mentioned just before that you have a part of the dissociation of the complex, and here on the sacs, you don't see any kind of dissociation of the complex with OD14. So what are the differences between the SEC-MALS experiment that you... I know you didn't present the SEC-MALS.
Yeah.
So, okay. Okay. But if the... Yeah. Because we have the result and we did the SEC-MALS. You can mention with the SEC-MALS we did the experiment and we see a kind of dissociation again that we don't see on this column, but the columns are not the same. This one is really tiny column that has not is not really resolutive, whereas the one that is used with SEC-MALS, it's bigger, and you have a higher separation of the different forms of the complex. So just to mention that not exactly, it seems to be the same experiment, but it's not really the same one, as the column are not the same in both experiments. Just on slide 14, what is this interaction map over 60%? What does it mean?
So it's highlight the context that appear in stimulation more than 15%.
Yeah, that's the interaction map. That's the contact that are present in more than 60% of all the simulation that she had. Okay. Mm-hmm. It's not 100%, so it's still an average, but because probably everything is moving, that's this 60% means that, and you have
why do we need to delete the first reason the graph to make this kind of map?
Because the intracyclic disorder part of Rev and glutamate, we know that it's probably moving a lot, and it's a lot of work and needs a lot of computer, how can I say, not energy, but it's a lot of
the RAM.
Sorry?
The RAM.
Yeah, it's a lot of I don't have the word to compile, and because each residue are moving, and so when the complex are too high, it's not feasible for the moment to do an molecular dynamics. So the idea is to do it only with the folded part as the beginning, and again you can also mention that you focus only on the folded residue, that the residue that are located on the folded part. At the beginning you mentioned me why we didn't perform the mutagenesis of residue, I don't remember, 115, for example, just due to the fact that this residue is located on the IDR, and we know that the IDR is always moving, so probably the location of this residue is here at one point, but it's moving too strongly, we can say, and that's true, that is not really feasible. So what is your conclusion from this, from your results based on this molecular dynamic simulation? What do you think about the result, and what are the conclusions that you can draw from this experiment?
Because the interaction is highly multivalent, and because RAP have the RNA-rich motif, so when we mutate this residue, it cannot use another precision on the RNA, the ARM to reach to the OD.
So where is located the arginine-rich motif on RAP?
I just show on here, but it's around here.
Yeah, it's on this alpha helix. So you can mention that the conclusion can be that we have several application of residue, and it's probably a combination of some of them, and by disrupting this, what we can say key points from this modelization, from this AlphaFold prediction, is not sufficient, and perhaps we have also a role of the N-terminal tail, and it's for that we want now to combine this mutation on the truncated form. But if we don't see any effect of this double mutation on the context of delta 14, we can also think about the fact that it's AlphaFold prediction now, and it's that the starting point is not really correct. So like this, you don't believe everything. You can just keep in mind that that's true, that's an AlphaFold prediction. Even if we have a high confidence of this prediction, we have to keep in mind that it's only
the
prediction, and it's for that we combine all the different experiments to try to answer to this question, even if we don't have any crystallographic or high resolution result on the complex. And so why you didn't test cryo-EM and X-ray crystallography to try to solve the structure of the complex?
About the result in the...
So the members of the jury that didn't read your report can ask you, why did you didn't perform X-ray crystallography and cryo-EM experiment to try to have a higher answer?
Because I did perform, but RAP is too flexible on the OD scaffolds, and we try to cross-linking to stabilize RAP on the OD to capture the clearly precision of RAP on the OD, but the cross-linking haven't finished, and this didn't work with the glutathione and BS3, so I didn't show it on here.
Okay, so you for the X-ray crystallography, you can say, okay, it's quite difficult with REV, with the N-terminal tail, the C-terminal anthracyclic disorder tail, and the truncated form is not really stable. So that's one answer. And so it was not the technique that we wanted to test in the current the internship. And then we started the cryo-EM, but we encounter several problems. The favor orientation of the molecule. We have something weird, and it seems to like the complex seems to be dissociated on the grids. So we started to work with cross-linking experiments, but the results obtained were not really good enough to go further. So that means that, yes, we started to work with, but we cannot present the result because of that. Okay?
Yeah.
From the NMR experiment, can you just precise on the graph which residue do you consider that are interacting with REV?
Which residue? We cannot predict exactly which residue. Just can because it's just it,
Can you really not predict, or do you have an idea? Do you think that the C-terminal part of the HBD is involved in the interaction?
Yes.
The C-terminal part of HBD?
The C-terminal is
no. This part.
No, is not in the. It's not in the interaction because when we add more draft, the intensity didn't change too much in this terminal.
So we can consider that it's more on that part, and even if here why do you don't have any attribution here?
Because this is containing the D and E, so this is acidic or acid.
Mm-hmm.
So the environment is the same. So at the first we cannot.
So we have a lot of acidic residue on that part, D and E, and it's located here.
Yeah.
And it's the peak are really difficult to dissociate. So we cannot really attribute them. We know that this part is moving, so seems to be involved in the interaction, but we cannot really know which peak are from which residue in a specific sequence. So so the ID seems to be more here. And can you explain that? Can you give what do we see on this kind of graph?
On the both two relaxation.
Mm-hmm.
So
so perhaps you should have a title on the relaxation. Yeah.
So what are you asking?
Can you explain this kind of graph, because I don't know about that.
First is when the graph show the R1 row ratio. So this data increase when the protein switch back and further between the free and bounded state, and because it's in intermediate speech. So first at the HBD, when we add 20% of graph, the first up to 100 increase, which mean that the APD is moving slower. So it's so
slower.
Yeah.
HBD is moving slower.
When
Are
we
you sure?
When we add graph,
it's much higher, no?
Because
it's per second, no? is not
What?
is the scale is not per second.
Per second, microsecond to millisecond.
Yeah, but it's showing here that is moving
Yeah.
much higher than without rev.
Yeah.
Okay.
This data increase, which means the moving is slower.
Okay.
The free state is
I have to ask to Elise because I understood that it was the moving was faster, and showing that the affinity is not so strong.
see explain with me that first with the relaxation. So if the protein free, this data is will lower, and when it's by with the rev, the moving is restricted. So if it's really have the interaction, the movement of the protein will restrict, so it's slower. And with the ODHVD, she said with me that it's extend to the slower extend in toward the slower.
So this one, the exchange is slower.
Yeah.
Showing that the affinity is perhaps higher when you have OD and HBD. But here, for me, is the illustration that there is a change between in absence or in presence of rev. That's the illustration that we have dynamic here with this residue. So I have to ask to Elise because it's not fixed like this on my mind. So okay, so I think it's good. It's much better than last week. So and even if I think you should try to
slower.
Not slower, but just changing the intensity of the voice. Okay, the rate is quite good, and you seems to be much less stressed than last week. So that's really a good point. But the level of your voice, it's like this. If you can, question 1. At the end of question 1, your voice has to decrease, and then take and now you start again with the question 2. Try to think about that, to slow down just at this point. Okay, so what I propose you is that you do a next rehearsal tomorrow, and we ask 2 or 3 people from the lab to come and to ask other question, and like this you will be on the top on Friday. So at what time? I didn't ask you if you agree or not. I don't want
in the
any
afternoon.
answer in the afternoon.
Yeah.
So we try around 2 again. I can send a message for that and ask who is available to come to listen you. It will increase a bit the stress for sure, but on Friday it will be so easy.
No.
Yes. For sure it will be much better than don't do any rehearsal on tomorrow. I think just to listen, because the people that will come, I hope that some will be available, but they don't know a lot about this topic. So it's good just to, if there is something that is not clear, probably not change the slide or minor things, but just say, okay, here it's not clear, so perhaps I can add these little things before, and just to, the idea is to try that all the people in the audience on Friday, without reading your manuscript, understand what you did. If it's the case, that's the top. Okay?
Yeah.
It's for that sometimes you don't have to add a lot of information, otherwise you lose the people that are currently listening. If you give some sufficient information, that they can follow all the story, and then they will ask questions about details that you didn't give them.
Yeah.
Okay? So I send an email for that, and we try to find some people available. So